Hey everyone, welcome! St. Paul American School Clark Grade 9 students. I am absolutely thrilled to be guiding you through today's explainer on G9 Biology, Unit 03, Lesson 02, Mendelian Genetics and Gene Linkage and Polyploidy. Today, we're going to look at the absolute foundational rules of heredity, and then, well, we're going to break them. It's a seriously fascinating journey into how traits are passed down, how chromosomes behave, and what happens when the genetic code does something entirely unexpected. Okay, let's dive into this. Here is our roadmap for today, which we can basically call the rules and the exceptions. We'll start with Mendel's laws, explore gene linkage and recombination, and map some genes. Then we'll zoom out to polyploidy, do a quick misconception check, and finish off with a recap. Let's get right to it. Section 1. Objective and Mendel's laws. The foundational rules of heredity. So our primary objective here is to really understand the basic rules established by a 19th century monk named Gregor Mendel. Between 1856 and 1863, Mendel cultivated and tested an astounding 28,000 pea plants. I mean, can you imagine that kind of patience? From all that work, he deduced two main pillars of classical genetics. First up, we have the law of segregation. This states that an individual possesses two alleles for a trait, but during the formation of sex cells, these separate. Basically, a parent passes only one of those alleles to their offspring. Second, we have the law of independent assortment. Mendel crossbred pea plants with different traits, like, seed color and seed texture, and found that the inheritance of one pair of genes is completely independent of the inheritance of another pair. But here is the critical caveat we know today. This second rule only holds true if the genes are on entirely separate, non-homologous chromosomes. Real quick, before we move on, let's just make sure we're on the same page with the essential vocabulary for genetic crosses. If an individual has two identical alleles for a trait, say two dominant capital A's or two recessive lowercase a's, they're homozygous. If the two alleles are different, so a capital A and a lowercase a, they're heterozygous. And remember, in a heterozygous individual, the dominant allele is going to hide the expression of the recessive one. So the recessive trait is technically there in the genotype, but it's totally hidden in the physical phenotype. Let's move to and see how this builds. Section 2. Gene linkage and recombination, the plot twist in Mendel's rules. All right, this brings us to a massive, critical question that challenges everything we just established. We know independent assortment works if genes are on separate chromosomes. But what if genes are located on the exact same chromosome? Well, the answer is genetic linkage. This happens when genes are located physically close to each other on the very same chromosome. Because they're essentially next-door neighbors, they tend to be inherited together. They travel as a package deal during meiosis. And you guessed it, this phenomenon is a direct violation of Mendel's law of independent assortment. They absolutely do not sort randomly. It helps to think of genes as being organized linearly on chromosomes, almost like beads on a string. If you have two beads sitting right next to each other on that string, wherever the string goes, those two beads go together. That's the physical reality of linked genes. However, as you'll see, biology always has a clever workaround. While linked alleles do tend to stay together, they aren't permanently glued. During the first division of meiosis, homologous chromosomes pair up and can actually exchange linear segments of genetic material. This is what we call recombination, or crossover. Basically, this process shuffles the maternal and paternal alleles around, which can separate linked genes and create brand new combinations in the offspring. We call these recombinant gametes. Section 3. Worked example. Mapping genes. How we measure genetic distance. It's pretty wild to think that something as tiny as a fruit fly actually helped us decode the map of life. Scientists Thomas Hunt Morgan and Alfred Sturtevant used Drosophila melanogaster, common fruit flies, to figure this whole thing out. Morgan analyzed traits like body color and wing shape, and he provided the direct, hard evidence that genes were linked on chromosomes. Then Sturtevant took it a step further in 1913. He created the very first genetic linkage map. He realized that the frequency of recombination between two genes could actually be used as a direct measure of how far apart they are. By performing highly specific test crosses, they literally charted the linear order of genes. And this brilliantly illustrates exactly how they calculated those distances. It's actually a pretty straightforward formula. First, you just count the number of recombinant offspring, the ones that are showing those new shuffled traits. Then you divide that by the total number of offspring and multiply by 100. That resulting percentage, that equals the map distance in map units, sometimes called centimorgans. So if you get a 12% recombination frequency, that means the genes are exactly 12 map units apart. The rule of thumb here is, the further apart two genes are on a chromosome, the more likely it is that a crossover event will happen right between them.
Section 4. Polyploidy in eukaryotes, when entire chromosome sets multiply. Now let's zoom way out from single genes and linear maps to some massive structural genetic changes.
So the term ploidy, great word, right, refers to the number of complete sets of chromosomes an organism has. Humans, along with most animals, are diploid, meaning we have two complete sets, one from each parent. But nature gets pretty crazy, and those numbers can go way higher. We can have triploids with 3 sets, tetraploids with 4, hexaploids with 6, and get this, even octoploids with 8 complete sets of chromosomes. Now, while polyploidy is incredibly rare in animals, though, fun fact, a polyploid rat was famously discovered down in Argentina, it's actually naturally occurring and just incredibly widespread across the plant kingdom. In a really fascinating study on Azalea breeding, we see just how common this is in the rhododendron genus. So how exactly does this happen? Well, sometimes there's a spontaneous doubling of chromosomes within a single plant just because of a cell division error. We call that an autopolyploid. Other times, unreduced gametes from two completely different species merge together, creating what's called an allopolyploid hybrid. Now, what's really interesting about this slide is the incredible evolutionary advantage this gives to plants. Polyploidy is a literal powerhouse for evolution. First off, it can cause abrupt speciation because it creates an immediate reproductive barrier. A new tetraploid plant often can't easily breed back with its diploid ancestors. On top of that, allopolyploids fuse two completely different genomes together, which drastically increases heterozygosity and hybrid vigor. They can exhibit this cool thing called enzyme multiplicity, meaning they produce enzymes from both parents, which grants them incredible biochemical flexibility to survive in totally new environments. And finally, just the sheer volume of extra DNA often physically enlarges the plant's cells, leading to thicker leaves and much larger flowers. That right there is exactly why horticulturists and plant breeders find them so valuable.
Section 5. Misconception and formative check. Testing our boundaries. We've covered a lot of ground today, and there's one specific hurdle in this unit that almost always trips students up.
Let's clear up something by contrasting independent assortment with genetic linkage directly side by side. You have got to remember that Mendel's independent assortment strictly applies when genes are on different chromosomes. That is why they sort randomly. Genetic linkage, on the other hand, is the exact opposite condition. The genes are on the same chromosome, physically close, and they travel together as a single inherited block. There are two completely different physical scenarios happening inside the cell. Okay, keeping all that in mind, let's put this to the test with a quick recall question. True or false? Linked genes can never be separated once they're on the same chromosome. Think back to the beads on a string analogy for a second. Got your answer? Actually, it's false. While they definitely tend to travel together, that process of recombination or crossover during meiosis can physically break that connection, shuffling and separating linked genes onto totally different homologous chromosomes. So the crucial point is, biology is built on rules, but it absolutely thrives on the mechanisms that break them. Section 6, recap of required concepts, bringing it all together. All right, here is your high-level summary of today's explainer. Number 1, Gregor Mendel established the foundational laws of segregation and independent assortment. Number 2, genetic linkage occurs when genes sit close together on the same chromosome, and this directly violates independent assortment, unless they get separated by recombination. Number 3, we can physically map the distance between these linked genes by calculating the frequency of recombinant offspring. And finally, number 4, polyploidy is a super widespread phenomenon, especially in plants, where an organism possesses more than 2 complete sets of chromosomes, driving evolution and creating major hybrid vigor. As we wrap up this explainer, I really want to leave you with a provocative thought to chew on. If polyploidy gives plants immense biochemical flexibility, thicker structures, and the ability to just abruptly evolve, could the intentional creation of artificial polyploids in our agricultural crops be the ultimate key to securing our global food supply against a rapidly changing climate? It's a massive, game-changing question, sitting right at the intersection of Mendelian genetics and modern biology. Thank you so much for joining me today, St. Paul American School, Clark Grade 9, and keep exploring these incredible genetic mechanisms.